The hypothalamus and the hypophysis are closely associated structures, and they function as the main regulators in charge of responding to internal and environmental stimulus in order to maintain the organism function within a narrow range of endocrine concentrations, which in turn permits maintaining homeostatic conditions. The hypothalamus is made out of several clusters of similar neurons called nuclei. Each nucleus is specialized in responding to different types of stimuli. The neurons of the different nuclei release specific neurohormones to regulate different physiological processes. The hypothalamus is connected to the hypophysis by the hypothalamic hypophyseal stalk. The hypophysis is divided into three main areas. The pars distalis, which is located cranially, and it is made out of 5 types of cells, each capable of responding to specific hypothalamic hormones and to secrete other hormones which will work in different processes within the body. The pars nervosa is located caudally in the hypophysis and, although does not produce any hormone, it receives axons of neurons located in the several nuclei of the hypothalamus. These neurons release their content into a capillary network which takes these hormones directly into circulation. In between these two structures is found the pars intermedia, which has a more limited function, but it is very important in animals living in desertic environments. A prolongation of the pars distalis into the hypothalamic hypophyseal stalk is the pars tuberalis, which supports the microcapillary network that makes the hypophyseal portal system. This microcapillary network is supplied by the superior hypophyseal artery. It starts in the median eminence and carries the secretion released by neurons of the hypothalamus through the hypophyseal stalk or infundibulum into the pars distalis. Within the pars distalis, again, this capillary bed is ramified to supply all the cells of the pars distalis. The pars nervosa is irrigated by the inferior hypophyseal artery and drained by the hypophyseal vein. The bundle of neuron axons traveling from the hypothalamus to the pars nervosa forms the hypothalamic hypophyseal neural tract. Neurons in the hypothalamus process the sensory information and determine the appropriate response to maintain homeostasis. That response could be carried out in 2 manners. Neurons of the hypothalamus can release stimulatory or inhibitory substances into the hypothalamic section of the portal system. They travel through these capillaries and reach specialized cells of the pars distalis, where they bind to surface receptors of these cells to either promote or to prevent the release of other hormones which, when released, enter into circulation and reach their target tissue throughout the organism. Neurons of the paraventricular and supraoptical nucleus synthesize ADH or vasopressin and oxytocin respectively. They travel along the axons of these neurons through the hypophyseal neural tract. Both oxytocin and vasopressin move within their respective neurons attached to neurophycins, which are large carrier proteins. These complexes are then stored in membrane-bound secretory vesicles called Herring bodies, which are located in the axon terminal of these neurons. Once they are needed, they are released into the capillary plexus located in the pars nervosa, and then they reach systemic circulation.